Hello, future doctors. I am Dr. Shivangi Shukla, and today we'll be discussing a very important topic in carbohydrate metabolism, and that is glycolysis. But we'll not just be discussing it, rather, we'll be breaking it down into very simple, easy to remember and understand steps, and therefore, we'll be making the whole process of learning about glycolysis very easy. So, how are we going to go about it? Glycolysis is a pathway involving the metabolism of glucose, which is a 6 carbon compound, breaking it down into two molecules of pyruvate, which is a 3 carbon compound. In this process, a lot of intermediates are formed and a lot of ATP molecules are utilized, are produced, and and NADPH molecules are also produced. Also, a few reactions are reversible, a few are irreversible, and there are a lot of enzymes which catalyze each and every step involving each intermediary. So, these are the four broad areas where we will be dividing this entire reaction, and we'll study each and every broad area. Then we'll incorporate each information that we are discussing into the final reaction pathway. Let's begin. This first slide I have purposely kept blank because after we read the entire pathway, we are going to come back and jot down the entire pathway by ourselves as we speak. So you keep a sheet of paper, a blank sheet of paper by your side. And just after our discussion, we are going to come back and using all the tricks, we are going to jot down what we have understood. So let's begin. These are the intermediates in this pathway. The first broad topic that we're going to discuss is the intermediates. What are the intermediates? Starting with glucose. Glucose is a 6 carbon compound. Okay, so one molecule, suppose we have one molecule of glucose. Okay, one molecule of glucose produces one molecule of glucose 6 phosphate, again a 6 carbon compound, which converts to a one molecule of fructose 6 phosphate, which is again a 6 carbon compound, which converts to one molecule of fructose 1,6 biphosphate, which is again a 6 carbon compound. Now, this 6 carbon compound breaks down into one molecule of dihydroacetone phosphate, which is a 3 carbon compound, and one molecule of glyceraldehyde 3 phosphate, which is again a 3 carbon compound. Now, each of these will then produce one molecule of bisphosphoglycerate. So, one molecule will come from this DHAP, one molecule will come from glyceraldehyde 3 phosphate. So, in all, we have two molecules of bisphosphoglycerate, which is again a 3 carbon compound. Then, these two molecules of bisphosphoglycerate will produce one each, that is, two molecules of 3 phosphoglycerate, again a 3 carbon compound. Then they will convert to one each of 2 phosphoglycerate, 3 carbon compound, converting to one molecule each of phosphoenol pyruvate, again a 3 carbon compound, and converting to two molecules of pyruvate, which is again a 3 carbon compound. So we are seeing that starting from glucose, which is the substrate, which is a 6 carbon compound, one molecule of glucose will produce two molecules of 3 carbon compound, that is pyruvate. Now, how do we remember these? These are very fundu sounding terms, but which can be remembered easily by a mnemonic. So I have created my own. You can use your own. This is GGFF. That is, you must have heard about the movie called Gone Girl, right? Gone Girl film features good dialogues. I'm sorry, I have interchanged. It's okay. Good or great dialogues by three people and two people for peppy party. So, Gone Girl film features good dialogues by 3 people and 2 people for peppy party. All right, so just remember this and you'll remember each and every intermediate. So we are through with the intermediates, and through this mnemonic, we can remember it sequentially in the correct sequence. That is very important. Next are the enzymes, which are very important. These enzymes I have written down sequentially as they catalyze each and every reaction. So there are a total of 9 reactions in glycolysis. The first one is catalyzed by hexokinase, the second by phosphohexose isomerase, then phosphofructokinase, aldolase, triose phosphate isomerase, glyceraldehyde 3-phosphate dehydrogenase, phosphoglycerate kinase, phosphoglyceromutase, enolase, pyruvate kinase. Everything is cluttered, everything is muddled up, but do not get scared. All these we need not ratify, we need not remember. There should not be the need for any mnemonics either. I'm going to break it down, make it very easy for you guys. Now, the enzyme kinase, wherever you find the enzyme kinase, it will involve the adan pradan or the give and take of phosphate group. Okay, so kinase, you hear kinase, and what should ring a bell in your mind is phosphate. There is some adan pradan going for about phosphate in this using kinase. So kinase is synonymous with phosphate. Hexo means 6. So a 6 carbon compound is getting involved with phosphate. That is, phosphate is getting added onto a 6 carbon compound. So a hexokinase catalyzes the conversion of glucose to glucose 6 phosphate. Got it? Phosphate is getting added onto glucose. Got it? So the first reaction is from glucose to glucose 6 phosphate. The 6 carbon compound, hexo compound, is getting kinased by phosphate. So glucose 6 phosphate. Second is phosphohexose isomerase. That means some 6 carbon compound which has phosphate on it is getting converted into its isomer. Very easy to understand. This is the 6 carbon compound containing phosphate getting isomerized into fructose 6 phosphate. Now you see this fructose 6 phosphate is additionally getting another phosphate group onto it, that is 1,6 biphosphate, right? Biphosphate, that is one more phosphate is getting attached. So if one more phosphate is getting attached, then which enzyme will come into play? Kinase. And how will we name this enzyme? This kinase is acting on this 6 carbon compound which has phosphate. So phosphofructokinase. Got it? Phosphofructokinase. This will fructose 6 phosphate to fructose 1,6 biphosphate. Got it? Next, aldolase. Aldolase enzyme converts this aldehyde into 2 compounds. That is, this fructose sugar, which is a ketone sugar. This aldolase enzyme will convert it into DHAP and glyceraldehyde 3-phosphate. Got it? Next, this glyceraldehyde 3-phosphate will then be converted into an acid, that is bisphosphoglycerate, by a dehydrogenase enzyme. Now, how is it so? I'm going to make it very easy for you. Glyceraldehyde 3-phosphate. This is an aldehyde. Got it? This is an aldehyde. Aldehyde will get oxidized to glycerate. Glycerate is an acid. Bisphosphoglycerate or bisphosphoglyceric acid. So this is an acid. This is an aldehyde. Aldehyde converts to acid by oxidation. So if this compound is getting oxidized, some compound will get reduced because oxidation reduction reactions are redox reactions. So aldehyde will oxidize to acid, and in the process NAD will get reduced to NADH. Got it? So addition of hydrogen is reduction. Therefore, aldehyde getting converted to acid is simultaneously accompanied by NAD getting converted to NADH. Got it? So this is the redox reaction. So glyceraldehyde 3-phosphate dehydrogenase. This hydrogen will go to NAD to form NADH. and this glyceraldehyde 3-phosphate will get oxidized to bisphosphoglycerate. Then that bisphosphoglycerate will be acted upon by this kinase to produce 3-phosphoglycerate, right? At the third position, phosphate has been added by kinase. This by mutase will convert to 2-phosphoglycerate. Then enol will be added, so therefore enolase will act. And after that, again phosphate will be added, so pyruvate kinase will produce pyruvate. Got it? So these enzymes are very easy to remember. Now they'll be even more crystal clear to you once we jot down the entire reaction by ourselves. Again, I'm saying we're going to jot down the reaction by ourselves. Got it? Next, to jot down the reaction, the third most important thing that we have to remember are the reversible and the irreversible steps. So the first, third, and the ninth steps are irreversible. Rest all are reversible. So for reversible, we have to make this arrow, a bidirectional arrow. And for irreversible, we'll make this arrow. Got it? So first, third, and ninth are irreversible. Next, the fourth most important step that we should remember is 2 ATP molecules are used, 2 NADH molecules are synthesized, and where are they synthesized? This we have discussed. And 4 ATP molecules are synthesized. Okay, so 2 ATP molecules are used, 4 ATP molecules are synthesized. So net 2 ATP molecules will be synthesized. This is for 1 molecule of glucose. So net production will be from 1 molecule of glucose, we will get 2 molecules of pyruvate. Got it? 2 molecules, 2 ATP molecules and 2 NADH molecules. Got it? So understood? We know that we know all the intermediates. We know the mnemonic, that is Gone Girl film features good dialogues by 3 people and 2 people for petty party. Got it? Then we know about the enzymes, that is kinase is used wherever phosphate is added or removed. Then isomerase is used whenever isomers are molecules are converting into isomers. Mutase is used whenever 2 forms are getting converted into each other. Then dehydrogenase is used whenever redox reactions are occurring. Okay, the hydrogen is going to form NADH. Then enolase will involve removing enol or addition of enol group. And yeah, that is it. So these are the basic enzymes that these are the basic properties of the enzymes that are involved. Then the next part was the reversible and the irreversible reactions. So 1, 3, and 9 are irreversible. Rest all steps are reversible. Then the total ATP molecules that are involved, 2 molecules are used, 4 are synthesized, therefore net 2 are produced. 2 molecules of NADH are also synthesized. Got it? So now every one of you must be ready. Now we'll come back to this blank slide. You guys go back to your own blank sheets and let it start. So the substrate was glucose, right? All of you, don't wait for me to write. You have to recall. So good, gone girl, right? Glucose. Now girl, that is glucose 6-phosphate. Film features fructose 6-phosphate features. So fructose 1,6-bisphosphate. Good dialogues, that means glyceraldehyde 3-phosphate. Dialogue is DHAP, dihydroxyacetone phosphate, by bisphosphoglycerate. 3 people, 2 people for petty Petty is PEP, petty, phosphoenol pyruvate. Party, pyruvate. Understood? Now the next thing was the reversible and the irreversible reactions. First was irreversible. Irreversible mein we are making this arrow. First, third was also irreversible. This is irreversible, and ninth. So this one is the ninth, the last one. These are the irreversible reactions. Okay, I'll use a different colored pen for you to be clear. Then the rest will be reversible. So let us make it like that. So they will also interconvert. Got it? Understood? Written down everything. Next, enzymes. Extremely important. Now, glucose converting to glucose 6 phosphate. Phosphate is getting added. Phosphate is getting added. Phosphate is getting added here. Phosphate is getting added here. And phosphate is getting removed here from phosphoenol pyruvate. Phosphoenol pyruvate में phosphate निकल के pyruvate बन जा रहा है. Okay. So whenever phosphate is getting added, whenever you have to attach something, you need energy. And whenever you have to remove something, you have to pull something out, you have to remove something, you don't need energy. So for attaching something, gathering something, okay, you need energy. So here ATP will be required. Okay. So whenever there is involvement of kinase, or wherever kinase is getting involved, ATP will be either produced or used. So here phosphate is getting added, so ATP will be produced, because you will require energy to add something. But to remove something, it is very easy. You just throw it away. You don't need any energy. But to add on to something, to gather something, you require energy. So ATP will be produced whenever phosphate is getting added. Okay, so ATP is getting produced here. This is the first step. Okay, remember we noted that 2 molecules of ATP will be produced. So this is the first step where ATP is getting produced. The next step where ATP is getting produced is when another molecule of phosphate is getting added here. Got it? So here also ATP will get produced. So 2 molecules of ATP got produced. Understood? Now here if you see, from phosphoenol pyruvate, we are forming pyruvate. So phosphate is getting removed. Here also kinase enzyme will be there, but phosphate is getting removed. So ATP, the stored ATP will be released. Got it? The stored ATP will be released. Yeah, so it's okay. So I will make these arrows. I have used these arrows should be like this, right? ATP is getting produced, ATP is getting used, right? Energy is getting used. So ATP will convert to ADP. Wait, I'll just, so that you don't get confused, I'll erase it and I'll make it again. Okay. So ATP is getting used here. ATP, that is energy, gets used. ATP gets converted to ADP. Here another molecule of ATP is getting used, that is ATP is getting converted to ADP. Understood? Energy is getting used in attaching phosphate. So ATP is getting used here. ATP is getting used here as well. Now here, just to remove phosphate, we don't require energy. So the energy that was contained in in attaching the phosphate, that energy will be released in the form of ATP. So you had a required energy to attach phosphate, and now when this phosphate is getting thrown away, that energy will then be converted to ATP and stored. Okay, now you might ask, we had talked about total 4 molecules of ATP. Where is the other molecule? Tell me, there must be some other reaction where ATP is getting involved. There must be some other kinase reaction. What is that kinase reaction? Let me know. This molecule of, okay, so we'll go one by one. So this okay, so we got to know about ATP that is produced here. Now, not to confuse you further, this one molecule of glucose will be broken down into one molecule of G3P and DHAP. Okay, so this is a 3-carbon compound, this is a 3-carbon compound, and they are interconvertible. So this reaction that is occurring with G3P, glyceraldehyde 3-phosphate, also occurs with DHAP. Okay, that is DHAP will again convert to G3P, and again another step will occur. So this will occur twice. This reaction, this series of reactions will occur twice. Once with this G3P and again with the DHAP, which has converted to G3P, will again go through this. Therefore, everything is occurring twice. Understood this? Now, this glyceraldehyde 3-phosphate will then convert to bisphosphoglycerate. This we knew how it is going to get converted, glyceraldehyde 3-phosphate, through the enzyme dehydrogenase. And how is it so? This dehydrogenase will remove hydrogen from glyceraldehyde 3-phosphate. It will oxidize it to bisphosphoglycerate, and NAD will get reduced to NADH. So here is the NADH molecule that is produced. Got it? Now, this bisphosphoglycerate molecule will then convert to 3-phosphoglycerate. And this is the step where another molecule of ATP is getting produced. This was what I had quizzed you. That is why I'm coming back to it, because we were skipping this NADH step and you could have got confused. Got it? So this bisphosphoglycerate converting to 3-phosphoglycerate and then to 2-phosphoglycerate. So this step is where another molecule of ATP is produced. So 1 ATP, 2 ATP, then again this DHAP will convert to this, then again 1 ATP, 2 ATP. So total 1 into 2, 1 into 2. So total 4 molecules of ATP are produced. Here 2 molecules of ATP are used. 1 molecule of NADH is formed, and then again DHAP, then 2 molecules of NADH will be formed. Understood? Got it? This whole reaction is so simple to understand. Now, if I quiz you, what is this enzyme converting phosphoenol pyruvate to pyruvate? Now, what should be your quick thinking? Phosphoenol pyruvate involves phosphate. Pyruvate does not involve phosphate. That means this enzyme must be some kind of kinase. And what is the important metabolite here? That is pyruvate. So what is the name of this enzyme? Pyruvate kinase. Understood? Now, what is this enzyme that converts G3P to bisphosphoglycerate? Here we see G3P is glyceraldehyde, aldehyde, converting to bisphosphoglycerate. That is bisphosphoglyceric acid, which is an acid. Aldehyde converting to acid is an oxidation reaction. This will involve a redox reaction of NAD converting to NADH. So there must be a dehydrogenase group involved. So glyceraldehyde 3-phosphate dehydrogenase. Got it? It is as simple as that. This whole reaction of glycolysis is broken down into 4 major areas. That is the intermediates, the enzymes, ATP and NADH involved and produced, and reversible and irreversible reactions. You can note it down on your own, and it is extremely easy. You do let me know if it made life simpler for you or you got more confused. Then we'll talk about it again in the next series. Thank you.